Good afternoon. Welcome to this week's Wednesday webinar. We'll just give everyone a couple of minutes to join before we get started. Welcome, everybody. I can see that we still have a few more people joining, so we'll just wait another minute or so before we actually get started. Good afternoon, everyone, and thank you for joining us for this week's National Hemophilia Foundation Wednesday webinar. My name is Fiona Robinson, and I will be your host today. In today's webinar, we will discuss a topic of great interest and excitement, gene therapy. Please feel free to ask any questions you may have throughout the presentation by using the Q&A feature at the bottom of your screen. We have NHF staff members monitoring these questions, which we will present to our speaker at the end of their presentation. This webinar is being recorded and will be available to the community beginning on Friday, June 16th. Today, I'm joined by NHF's very own Dr. Len Valentino. Dr. Valentino is the NHF President and CEO, and his presentation today is on gene therapy for hemophilia. First, do no harm. Thank you for joining us today, Dr. Valentino. I'll now turn things over to you.
Thanks, Fiona, and welcome everybody for joining us today. I wanted to present this presentation on hemophilia gene therapy. First, do no harm. So these are my disclosures. I have nothing to disclose, and the opinions expressed in this presentation are my own and not necessarily reflective of those of the National Hemophilia Foundation or its board of directors. The mission of the National Hemophilia Foundation is shown on this slide, and you see it both in English and in Spanish. And our goal for having both English and Spanish versions is to increase our ability to engage with the Spanish-speaking community, to enhance our ability to deliver education and advocacy efforts, and also involve more people in our research efforts. The National Hemophilia Foundation is dedicated to finding cures for inheritable blood disorders and to addressing and preventing the complications of these disorders through research, education, and advocacy, enabling people and families to thrive. So it's very important that we stay true to our mission when we're thinking about hemophilia gene therapy. The overview of my presentation is shown here. I'll break this into three parts. First, an introduction and the treatment of hemophilia. Second, I'll provide some information on hemophilia gene therapy and other innovative therapies for hemophilia. And last, I'll discuss preparing the hemophilia community for gene therapy. I want to emphasize that this topic is the subject of a review article that was accepted for publication by the Journal of Thrombosis and Hemostasis on Monday, June 12, 2023. This manuscript is a culmination of work that includes participation of multiple different authors, and you can see the list of authors on this slide here, including NHF's own Dawn Rodolini, NHF's Chief Operating Officer. I think it's also important to recognize, and you'll see in a subsequent slide, the important work that was done by our public policy team in terms of the submission of a citizen petition in 2022 to the Food and Drug Administration. That was really the lightning rod for the work that I'm going to discuss today. So I'd like to acknowledge and thank the co-authors, as well as Nathan Schaefer and our public policy team, for helping craft the contents of this presentation. So as you all know, hemophilia is a lifelong inheritable bleeding disorder due to the deficiency of the activity of coagulation factor 8 in hemophilia A or coagulation factor 9 in hemophilia B. Living with hemophilia has a significant impact on health-related quality of life due to its impact on frequent bleeding events, the economic burdens associated with healthcare resource utilization, and mental health burden. Prevention of bleeding with medications derived from human blood plasma or created by recombinant DNA technology are highly effective, but extremely burdensome and can be very expensive. So when we think about hemophilia, for decades, the goal has been to reduce the burden of treatment, the burden of disease, while also enhancing the efficacy of treatment to enhance the quality of life for people living with hemophilia. And I want to emphasize that the important work that's been done in terms of innovation in gene therapy for hemophilia really represents the tip of the iceberg, the beginning of addressing inherited bleeding disorders, including rare and ultra-rare bleeding disorders and von Willebrand disease, all of which the same health-related quality of life issues, the economic burden and healthcare resource utilization, along with mental health burdens, are even more important and detrimental to people living with those diseases. So I want to make sure that what we're learning for hemophilia is able to be translated into these other bleeding disorders and consistent with the mission of the National Hemophilia Foundation to be addressing blood disorders. That these are lessons that we can learn and then apply to other blood disorders as well to improve the health-related quality of life for all people living with blood disorders. So let's turn our attention to the treatment of hemophilia. The care, treatment, and support provided for people with hemophilia over the past 5 decades has steadily advanced, resulting in improved health outcomes in countries with economic capacity to afford them. It's important to recognize that hemophilia treatment centers have provided the medical home for people living with hemophilia, von Willebrand disease, rare and ultra-rare bleeding disorders, but also other blood disorders, including Glanzmann thrombocytopenia, sickle cell disease, and thrombotic disorders, just to name a few of the other blood disorders that are all cared for within our hemophilia treatment centers and the Hemophilia Treatment Center network. In the early 1990s, hemophilia was identified as an ideal test case for gene therapy because it's a monogenic disorder with a clearly defined phenotype and a wide therapeutic window. The hemophilia community has long awaited the availability of a cure which would provide optimal outcomes while alleviating the burden of treatment with a safe, one-time administered product. I believe that we're on the cusp of seeing this cure come to people living with hemophilia. So what are the treatment options that are currently available to people living with hemophilia? In this slide, I try to depict what the potential options are, because as we think about choosing hemophilia gene therapy, we must put that in the context of a shared decision-making framework. And it's important for people who are making this decision, people living with hemophilia, their loved ones, and people who care for people living with hemophilia, all have the necessary information to make an informed decision. So the first thing that we recognize here is that there are factor replacement treatments that are to the left, and you see that those represent standard half-life therapies, extended half-life therapies, as well as bypassing agents. The bypassing agents are primarily used for people living with hemophilia complicated by inhibitors. To the right-hand side of the slide, we see the non-replacement therapies. The first is enhancing coagulation. The prototypical drug for this is emicizumab, or the bispecific monoclonal antibody, which has the trade name Hemlibra. We also have several rebalancing coagulation agents in clinical development, including drugs like fitusiran, concizumab, and other tissue factor pathway inhibitor agents. Desmopressin is an important adjuvant therapy, which is a primary treatment for those people living with mild, in some cases of moderate hemophilia A, as well as von Willebrand disease. Aminocaproic acid and tranexamic acid are important antifibrinolytic therapies that are useful to help control oral bleeding, as well as other forms of bleeding, and are important adjuvant therapies to enhance the coagulation effects of factor replacement therapy, as well as in the non-factor replacement therapy arena as well. We'll hear more about those as we progress. So let's think about innovative therapies. I mentioned the bispecific monoclonal antibody, emicizumab. We also know that a new bispecific monoclonal antibody, Mim8, is in clinical development under the direction of Novo Nordisk. Bispecific monoclonal antibodies offer several advantages, including subcutaneous delivery, infrequent administration, oftentimes every week, every 2 weeks, or even once monthly in some situations. The negative of the bispecific monoclonal antibodies can be their inability to be monitored, as we do with factor VIII therapies or factor IX replacement therapies, as well as the possibility for the development of anti-drug antibodies. We've also seen the introduction of more potent factor molecules into the development arena. One of these we'll talk about in a second is efanesoctocog alpha, which is a more potent version of factor VIII molecule. The rebalancing agents that address activated protein C or tissue factor pathway inhibitor, again, are important new therapies that may be coming to market if their clinical development programs prove successful and safe. And of course, the topic of today's presentation, gene therapy. So let's talk a little bit about bispecific monoclonal antibody therapy, and in particular with emicizumab. And this is data that comes from a review article by Callahan and colleagues that was published in Blood. And what we see here are the aggregated results from the Haven clinical development program. This program ran over several years and included hundreds of people who received emicizumab in clinical development. The two bars are shown, and what we see here are individuals who had zero reported bleeding events or treated bleeding events in the dark blue, and 1 to 3 treated bleeding events in the light blue. What I want to emphasize is that emicizumab has an excellent effectiveness profile. However, we are not necessarily achieving the goal of zero bleeding. And people continue to have 1 to 2 bleeds, oftentimes 3 bleeds per year. And we're hoping that we can achieve better results with additional improvements in bispecific monoclonal antibody therapy. I won't minimize, however, the important impact that a drug like emicizumab has had for the hemophilia A community, in particular those living with inhibitors, where emicizumab has been transformational, allowing these individuals to live a more normal life. It's important to recognize, however, that we do have more room for improvement. And the clinical development program for a drug like MIM8, as I said, is a second generation bispecific monoclonal antibody, is hoping to deliver more potent effects. And this molecule has been estimated to be possibly 20 times more potent than emicizumab, which offers the possibility of lower doses while still maintaining equal or higher efficacy to emicizumab. Because of the extensive mutational development of this molecule, the possibility for anti-drug antibodies has also potentially been reduced. So we're seeing that there's some potential advantages to a second generation bispecific monoclonal antibody. The clinical development program remains to be concluded, and we'll see results of those hopefully soon at the International Society on Thrombosis and Hemostasis conference that will be held in late June, and as well as at the American Society of Hematology meetings at the end of 2023. So more to come on bispecific monoclonal antibody therapy. I'd like to talk for a brief moment about efanesoctocog alpha. This is an important drug for the treatment of people with hemophilia. This is a molecule that was engineered to have a prolonged half-life while also potentially providing a safe means of replacing factor VIII. So what you can see in the left side of this panel is the molecular diagram showing this molecule. This is a molecule that's built upon factor VIII Fc, and it's been enhanced by adding several additional components to the molecule to further prolong the half-life of factor VIII Fc over that of the base molecule. You can see on the right-hand side the comparative data in pharmacokinetic studies in which this molecule demonstrates a more prolonged half-life, such that by 1 week after administration, participants in the clinical development program were able to maintain factor VIII levels above the recommended 3 to 5%, according to the World Federation of Hemophilia third edition treatment guidelines. And in many cases, individuals were able to maintain factor VIII levels above 7 to 10%, where we believe they'll be able to suppress bleeding to a large degree. Again, this is a molecule that it's hoped will have a very low immunogenicity profile. And in the clinical development program, that was borne out. I think we'll see that in the real world data, hopefully that will continue to be the case, but we'll await more data as more individuals use this now that this has been approved for use commercially. This slide depicts some of the rebalancing agents. On the right-hand side, you see the schematic for the intrinsic pathway, as well as the extrinsic pathway of coagulation, which both feed into the common pathway. There's several opportunities for rebalancing agents to impact the coagulation mechanism. We can have molecules that are involved in tissue factor pathway inhibitor, as well as inhibiting activated protein C, or by inhibiting antithrombin, as well as protein C. On the left-hand side, you see these molecules represented. So we have anti-tissue factor pathway inhibitors. 3 are listed here. The one that is most further along in the clinical development path is concizumab, but we have others that are available that have different modifications and may have different safety and efficacy profiles. The siRNA or silencing agent that directs its attention to antithrombin, fitusiran, is also in clinical development. One of the advantages of fitusiran is subcutaneous administration, but infrequent as every 2 months. So we see that the anti-tissue factor pathway inhibitors, along with the siRNA, offer the advantages of subcutaneous infrequent delivery. Similarly, SERPIN-PC molecule addresses the opportunity to inhibit activated protein C as a means to enhance coagulation. There's also a molecule in clinical development that it's a protease nexin 1 inhibitor, which inhibits factor 9A. So we have multiple different opportunities to rebalance the hemostatic mechanism. And as people are considering a gene therapy solution to their care, they must put this into context of the factor replacement therapies, monoclonal antibody therapy, as well as these rebalancing agents, because the pros and cons, the efficacy, risks, and safety profiles of each of these should be considered as one is thinking about a gene therapy approach. So let's turn our attention to the topic at hand, gene therapy. So how does gene therapy work? The gene therapy that we're talking about for hemophilia involves in vivo gene delivery. This is a gene therapy product that's administered intravenously to the recipient through a peripheral IV. The gene therapy product, or AAV vector, adeno-associated viral vector, which contains the instructions for making factor VIII or factor IX within a viral capsid or shell to protect it from degradation while it's in the circulation, but also allows the vector to home to its target site, the liver hepatocyte. So the product is injected intravenously, it circulates in the plasma, and then is directed to the liver, where it binds to and is incorporated into the hepatocyte. Once it binds to and is delivered to the hepatocyte, we don't really understand what exactly happens at that point, but we do know that the viral capsid or the shell is degraded, and the DNA is incorporated into the hepatocyte, reaches the nucleus, where the instructions are then available for the liver cell to make the necessary factor VIII or factor IX protein. So this occurs in the liver hepatocyte. The factor VIII or factor IX is then synthesized and hopefully gets out of the liver cell and back into the circulation to be able to effectively treat the hemophilia A or hemophilia B that the patient has. When successful, hemophilia gene therapy offers the opportunity for a different quality of life for people living with hemophilia A or B. Some of the challenges are, well, what happens if the intravenous product is neutralized once it's in the blood? And this can happen when the recipient has neutralizing antibodies directed against the adeno-associated viral vector. And these antibodies will neutralize the vector and clear it from the circulation, precluding the vector from reaching the liver and being able to do its work in the hepatocytes. So neutralizing antibodies are an important impediment to successful gene therapy for hemophilia. So the gene therapy options that are available to treat hemophilia A and B, in 2022, the European Medicines Agency granted conditional marketing authorization to an AAV5 gene therapy product, an adeno-associated viral vector AAV5 gene therapy to treat hemophilia A. And this product is still under consideration by the U.S. Food and Drug Administration to treat hemophilia A for people living in the United States. However, in the United States, the U.S. Food and Drug Administration granted approval to another AAV5 gene therapy product to treat hemophilia B. This product was also granted conditional marketing authorization by the European Medicines Agency in early 2023 for people living in Europe with hemophilia B. So we have two gene therapy products, one to treat hemophilia A and one to treat hemophilia B. The hemophilia B gene therapy product has received approval in the United States and in Europe, while the hemophilia A gene therapy product only has received marketing authorization in Europe and remains under evaluation in the United States. I want to point out that both of these products are delivered by an AAV5 capsid vector. It's important that this AAV5 mechanism is the delivery construct that's been used for both of these gene therapy products. They have different genes within them, but the viral shell is essentially the same. So to summarize the clinical trial data, in hemophilia A, those individuals who received valoctocogene roxaparvovec, the year 1 data showed a decline in factor VIII activity. However, many of these individuals had sustained levels over 2 and some over 3 years. However, there is a consistent decline in the factor VIII activity with time. Following a single dose of etranacogene dezaparvovec, participants with hemophilia B were observed to have stable factor IX levels for up to 3 years following dosing. So the pattern is somewhat different for those with hemophilia A who received the hemophilia A gene therapy product versus those who received the hemophilia B gene therapy product. Currently, the efficacy data for both valoctocogene roxaparvovec and etranacogene dezaparvovec both demonstrate similar impacts on bleeding rates compared to factor replacement therapy. So what you can take from this is that both products are very effective at preventing bleeding. They're both, in the short term, demonstrated to be very safe, although the use of steroids to prevent the immune-related complications of the AAV5 vector were evident in clinical trials for both products. The long-term safety we'll talk about in a few minutes. So it's clear that both products are effective at reducing bleeding and are relatively safe in the short term. So when we think about the efforts to protect patients from harm, in 2022, the National Hemophilia Foundation submitted a citizen petition to the United States Food and Drug Administration requesting that a risk evaluation and mitigation strategy, otherwise known as a REMS, be required as a condition of approval for both valoctocogene roxaparvovec and etranacogene dezaparvovec. Other organizations, including the World Federation of Hemophilia, the Hemophilia Federation of America, and the European Hemophilia Consortium, joined this effort by submitting separate but supportive statements to the U.S. Food and Drug Administration. In some cases, especially when there are unknowns, additional risk mitigation measures beyond the prescriber information that's contained in the package insert may be necessary to protect patients from potential harm. Now, why would we consider a citizen petition and a request for a REMS be implemented by the Food and Drug Administration? So the thought process is shown on this slide. When we perform a risk analysis for a novel therapy, we think about several different categories in this analysis. We think about what we know we don't know, what we have forgotten we knew, and what we don't know we don't know. And this framework is taken from an article that was published by Pierce and Iorio several years ago, but provides a very important way to think about risk in gene therapy. So let's look at the upper left-hand corner first. What we know. These are the knowns. So as I mentioned earlier, we know from the clinical development program for both hemophilia A and hemophilia B gene therapy that steroids are necessary to control the immune response for most participants who are receiving these products, especially the hemophilia A gene therapy product. Steroid toxicity is something that is very well known and documented and was observed in the clinical development programs for both products. Transaminase elevations or elevated liver enzymes was also something that was observed in both clinical development programs, more so in the hemophilia A gene therapy program than in the hemophilia B gene therapy program that we mentioned. There were also peri-injection reactions after the gene therapy product was administered, sometimes a flu-like illness, often accompanied by fever and chills. These are infusion reactions that we have to recognize are possibilities. Now let's look at the upper right-hand corner, what we know we don't know. We don't know the significance of the elevated liver enzymes during that first year. In the past, we've seen this scenario play out. 25 years ago, we encountered individuals who had something that we called non-A, non-B hepatitis, later shown to be hepatitis C due to the virus that causes the hepatitis C in the liver. These individuals had low-grade liver inflammation and minimal elevations of their liver enzymes, and we didn't think much of those elevated liver enzymes. However, we now know that elevated liver enzymes that were present during the early stages of hepatitis C were the harbingers of later complications. So what we don't know is what is the significance of the elevated liver enzymes that were observed in the first year and sometimes even later liver enzyme increases were observed. So we don't know the significance of these. Another important area are the consequences of vector integration. 20 years ago, we thought about AAV as an integrating vector. We sort of forgot about that for a while, but we don't understand what the consequences of vector integration are. And when quadrillion particles of vector are administered, even if the integration rates are only 1 in a million, that results in millions of vector integrations into the host genome. What are the consequences of that? We're not exactly sure at this point. Could there be unknown consequences into the future? Possibly. I think it's going to require long-term follow-up of individuals who receive gene therapy for hemophilia A and hemophilia B, as well as for other non-hemophilia related diseases that are treated with AAV vector gene therapy. Let's look at the lower left-hand corner, what we forgot we know. So these are the unknown knowns. We forgot that AAV vector integrates. We knew this 30 years ago. We forgot it after that, but we've realized that again currently. AAV vectors are predominantly non-integrating, but they do integrate at low rates. somewhere between 1 in 100,000, maybe 1 in a million, but that integration, if into a very sensitive site in the host genome, could have unpredictable effects. We also know that there's the possibility of acute liver injury. We've seen this and observed it in several gene therapy trials with AAV vectors for diseases outside of blood disorders, for non-hemophilia gene therapy. These are important observations that are present in some of the muscular dystrophies, as well as muscle diseases, where acute liver injury has been serious and in some cases has been fatal. So we need to take that into our risk analysis as well. And then in the lower right-hand column, these are what we don't know, we don't know. So it's very hard to predict the future. So what we don't know include areas and issues like thrombotic microangiopathy. Before gene therapy was administered, we became familiar with this term when TMA, or thrombotic microangiopathy, was observed when patients and individuals received emicizumab in the context of bypassing agents, and TMA events were observed. It was determined that those TMA events were most likely related to complement activation as a result of the bypassing agents that were administered. So thrombotic microangiopathy has also now been observed in gene therapy trials. So this is an important component of our risk analysis. Complement activation and the consequences of complement activation are also important risks to be considered. Dorsal root ganglion toxicity, or a nerve toxicity, has been observed in several non-hemophilia gene therapy trials, primarily for neurological and neuromuscular diseases. Will that be the case also in hemophilia? Remains to be determined. Hemophagocytic lymphohistiocytosis is a very rare complication of gene therapy. Again, not observed in the hemophilia trials, but has been observed in other AAV gene therapy trials. And of course, what could be next? We don't know what is lurking that is beyond our limit of detection currently. And only after more individuals receive gene therapy will we really be able to understand what the true unknowns are. So as we think about what is the goal for people living with hemophilia, well, first, it's important. We want no bleeding. So these products must be effective and efficacious in preventing bleeding. We'd like to ensure normal mobility so that the joints can be rehabilitated and that we have the ability to participate in leisure and sports activities for people who live with hemophilia. We'd like to have normal hemostasis so that individuals can undergo procedures and surgeries without being fearful of not having the hemostatic capabilities. And of course, health equity. And this is a wonderful example of health equity. Now, not every person with hemophilia aspires to be Chris Bombardier and scale Mount Everest, but it's possible. And Chris is a great example of what can be accomplished with appropriate therapy for people living with hemophilia. We'd like this to be the goal for people throughout the developed world, but importantly, also in the developing world. So I'd like to give a couple of final remarks. And this is a slide that comes from my colleague, Mark Skinner. And what is hemophilia, the aspiration for hemophilia in the developed world? We'd like to see optimized health and well-being or health equity. We'd like to see the prevention of joint disease and the prevention of bleeding. And this can only be achieved when we can maintain factor 8 or factor 9 activity levels above a minimum of 3 to 5%, as recommended by the treatment guidelines of the World Federation of Hemophilia. And this can be currently achieved by regular factor replacement therapy or substitution therapy with monoclonal antibody therapy like Hemlibra. You see the diagram from the manuscript on the right-hand side, the ascending staircase towards health equity. And as each of these milestones is reached, the individual has a greater likelihood of having health equity. This is clearly not the case in the developing world, where health resource utilization is not the same as in the developed world. We strive to have health equity, not just in the United States and in Europe and Australia and other parts of Western Europe. We would like to see this be a worldwide health equity so that the developing world is no longer disadvantaged because of living with hemophilia. So in closing, many people with hemophilia will choose not to receive gene therapy at this time for a variety of reasons, including the complexity and uncertainty of gene therapy products and the required follow-up. The unknown effects of gene therapy in both the short and long term are also likely to impact decision-making. The psychosocial impacts of gene therapy must also be considered, as the loss of identity and what's been termed the burden of normalcy as people with hemophilia transition to a symptom-free life may weigh heavily on some people with hemophilia. So as we prepare the hemophilia community for gene therapy, we must first train and educate healthcare professionals on hemophilia gene therapy. Similar training must occur for all stakeholders on shared decision-making, and this includes our healthcare professionals, people with hemophilia, and the caretakers and loved ones of people with hemophilia. The facilities administering either hemophilia A or hemophilia B gene therapy should be experienced and potentially be certified in the administration of the gene therapy product. Federally recognized hemophilia treatment centers should be the sites of care for hemophilia gene therapy, as they have the knowledge and expertise in evaluating, administering, and managing people with hemophilia. They're also the sites where the clinical trials for hemophilia gene therapy were conducted, and therefore they have the knowledge and experience to both evaluate the individuals, but also provide the short and long-term care to optimize outcomes for people who choose to receive gene therapy to treat their hemophilia. And finally, individuals who choose to receive gene therapy should be enrolled in a registry to collect robust data, including adverse events of interest. I believe that this is critically important, that we have a global, worldwide registry, such as that's being developed and has been put forward by the World Federation of Hemophilia, the World Gene Therapy Registry, to be able to collect the important data to demonstrate any early signals that are harbingers of safety in the hemophilia gene therapy products. So with that, I'd like to thank you for your attention, and I think that we have a few minutes to open up the panel for questions and discussion. Thank you.
Super. Thank you very much, Dr. Valentino. That was super interesting. We will go ahead now with the live Q&A. We do have a few questions that came in from our audience during your presentation. And also a reminder to our audience, if you have a question for Dr. Valentino, please enter it in the Q&A feature at the bottom of your screen, and we'll do our best to answer all your questions. So perhaps I can start with one question that did come in. And I think you've kind of addressed it, but it's this question about, what has been approved by the regulators, the FDA and the EMA. It's not quite the same. Can you just recap again what's been approved by them, and if you have any thoughts about why that's not the same?
Sure. So first, it's a good question. So the hemophilia A gene therapy product, Roctavian, as it will be called, has been approved or has conditional marketing authorization by the European Medicines Agency for treatment of people with hemophilia A in Europe. the U.S. Food and Drug Administration is still evaluating the risks, benefits to this product for people living in the United States. It's anticipated that a decision will be reached probably in the next several weeks, potentially, and that marketing authorization could come as soon as the end of June. The reason that there's a discrepancy is that the evaluation criteria, although harmonized, are not identical. The reviewers may value different attributes of the products in terms of safety as well as efficacy. And it's been clear that the Food and Drug Administration has asked the developer of Roctavian for additional data on their 2 and 3 year results. So I think that the request for additional data is most likely related to the decrement in factor VIII activity that's been observed. But I'm fairly confident that this product will achieve approval in the US in the not too distant future and be available. I think it'll be an important addition to the treatment armamentarium for people living with severe hemophilia A in the United States. Now, both the European Medicines Agency and the US Food and Drug Administration have granted authorization for hemophilia B gene therapy, etranacogene. And again, the reasons why those are similar, and in hemophilia A, it's dissimilar. It's not entirely clear why that's the case, but we have to rely on the expertise of our regulatory agencies to protect the safety and health and well-being of the populations that they serve.
And I think you've just answered that, but we had a sort of a follow-up question on that. Do you feel that the providers should wait for the regulators to provide that approval, because this is such an exciting advancement and there has been a lot of excitement during the wait for the approval? We have somebody asking, why should providers still wait?
Well, I think providers have to wait. If you live in the United States, you don't have access to hemophilia A gene therapy because it's not approved by the Food and Drug Administration for use. Participants who wish to join a clinical trial could access gene therapy through the clinical development program, but not commercially. So providers have to wait for hemophilia A gene therapy in the United States. It is available for hemophilia B. However, at least to my knowledge, there has not been a hemophilia B gene therapy patient treated with commercial product yet. And that may stem more from the reimbursement issues more than the regulatory issues. I think it's important to recognize that the reimbursement path is not entirely clear at this point, and I think there's still some questions regarding reimbursement for hemophilia gene therapy.
Thank you for that. There's been another question that's come in around the word cure. This is a word that you used at one point, and it does get used a lot when we talk about gene therapy. And this person is asking whether you believe that cure is a term that we should use for gene therapy, or if this is more personal to the individual.
I firmly believe that it's individual applicable. An individual is the only person who can truly define what a cure means for them. So some people have told me that a holiday from factor replacement therapy would be considered a cure for them. And if they had 2, 3, 4 years of not having to do regular injections of factor replacement or subcutaneous injections, they would consider that a cure, even if they had to go back to doing their regular therapy after some period of time. Other people have said A cure will only happen when my daughters are not at risk for having hemophilia. So, we're decades away from germline editing. So a true cure for hemophilia in which the germline would be modified so that a man could not pass hemophilia on to his daughters, that is decades away. And there's currently an international moratorium on germline editing. So this kind of research is prevented or precluded from occurring. So I think if you want to go to the extreme of what cure might be defined as, we're a long way away from that. We talk more about functional or effective cure, and that might be attainable with hemophilia gene therapy today with the current methodology, where an individual may have 5, 10, maybe 20 years of effective factor levels without having to do regular replacement treatment, whether that's with factor concentrates or monoclonal antibody or rebalancing agent. So I think it is a very individualized and personal definition of a cure, and only people that live with these diseases, our lived experience experts, can truly define what a cure is for them.
Thanks. We've had a couple more questions come in just now. One of them is asking, they said that they believe they saw concizumab has been approved for use in Canada. What do you think the prospects are for approval by the FDA?
Again, I think the clinical development program has been shown very promising results, both in terms of safety as well as efficacy. I think the likelihood is high that concizumab will be granted approval in the United States. And I think it potentially is yet another important therapeutic option that people will need to consider when deciding what is the optimal therapy for them. The advantages of a drug like concizumab may be an advantage for one person, may be a disadvantage to another. And what I'm speaking about is it's a subcutaneous delivered molecule, but it's delivered once a day. So that may be a disadvantage for somebody who's looking for once a week, once every 2 weeks, or once a month therapy. But others may consider once a daily therapy a sense of security, knowing that I've done my injection and I know I'm protected. So I think it's, again, going to be a very individualized approach, and that shared decision-making model process really needs to be implemented in our hemophilia treatment centers. And more importantly, people that live with hemophilia must embrace and demand a shared decision-making approach when they see their healthcare professionals. So as a person living with hemophilia, you need to be an active participant in that decision-making process.
Thank you. Another question's come in around the cost of gene therapy. If you'd like to venture a guess as to what the cost of gene therapy will be and if it will be covered by private insurance.
So the simple answer is no, I don't want to venture there. I think this is a really challenging question. There is no doubt that this is a highly innovative therapy, and the development of innovative therapies is very expensive. We know that the technology, the production, and the clinical trials are all extremely expensive. So when a company goes into the development of an innovative therapy, their research and development, their clinical development program, all contributes to a very high cost for that product development. And of course, companies are all for profit, and they have a fiduciary responsibility to their stakeholders, which are their shareholders. And many of us rely on those companies, as part of our retirement plans and 401ks and 403bs and whatever you rely on for your retirement, you want to ensure that companies are profitable because your retirement depends on those. So the cost of these products is commensurate with the cost of development. Do I think that it's necessary to have a $3 million price tag for a single dose of a product? Who am I to make that determination? I don't live with hemophilia. I don't know whether that cost would be justified to a person with hemophilia. And again, I think like the decision to choose to take a gene therapy product, it has to be very individualized. And is that cost worth it to you as a person living with hemophilia? Only you can make that decision. I think our payers are looking at gene therapy and are looking at what the overall cost of that's going to be to their bottom lines. But I think we've heard from most payers that they're considering paying for gene therapy because of certainly for hemophilia B, the durability seems to be there. For hemophilia A, I think payers are a little bit more skeptical, but I am confident that hemophilia A gene therapy will be paid for as well.
This is a particular question that's just come in asking whether gene therapy would qualify for vaccine injury compensation fund if there is a serious adverse event or injury.
Yeah, that I have no information on.
Okay. And one last question that did come in as well, and this is asking about the citizens petition that you talked about, whether you had any reaction or response to it from the community or from the group that you were petitioning.
Yeah, so the petition is submitted to the Food and Drug Administration. They are not obligated to reply or to make any comment on the citizen petition. They can choose to implement the recommendations in the citizen petition. In the case of the hemophilia B gene therapy product, they chose not to implement those requests. It remains to be determined whether they will consider our request if a hemophilia A gene therapy product is approved. So as I said, they're not required or obligated to make any correspondence or comment back to the petitioner, only implement the recommendations in the event of an approval. And at least in the case of the hemophilia B gene therapy, that was not the case, but it remains to be determined if in the hemophilia A situation, it will be adopted or not. So it remains to be seen.
Excellent. Okay, well, thank you very much. Those are all of the questions that we've received, so thank you very much for that. I'd like to thank Dr. Valentino for taking the time to join us this afternoon. We appreciate your time and expertise. I'd also like to thank all of you for joining us. This recorded webinar will be available on Friday, June 16th at hemophilia.org under the Events tab. If you select past events, you'll find all of our archived webinars there. Also available in the Events tab is the schedule of upcoming webinars in this weekly series. Please note that the Wednesday webinars will take a break for the next few weeks as people travel to and attend the International Society on Thrombosis and Hemostasis meeting, or ISTH, as Len mentioned, and then for the July 4th holiday. The next Wednesday webinar is scheduled for July 12th. Thank you very much for joining us this afternoon.
Thanks, everybody.